I was thinking like how to introduce this. I've seen that many of you coming to visit this UGM are more on the chemistry side of things. So I thought I'd start off with a chemical representation of a molecule. This is Cyclosporin A. I just brought it up because, you know, antibiotic development is recently a hot topic. Cyclosporin is what is actually produced naturally. If you are interested in getting more information about cyclosporin, like you can go to ChemSpider or PubChem and find additional biological data to this compound. If you're interested, however, in the structure, you can use our name to structure utility and just type cyclosporin and it pops up in Marvin. That's beautiful. However, if you're not quite sure about or using ChemDraw, for example, you might just have a chemist draw this for you. The question is, what is that molecule? Like you see this is a quite complex chemical structure, if I may say. If I go and say, I have an offer for an alternative representation, which might actually be a little bit more appealing for, say, a biologist, this is also a valid representation. Essentially, you can see this is a peptide. It's a cyclic peptide. It contains a number of amino acids, 11 in total. And if you look at the individual residues, you might see that actually none of these, or almost none of these amino acids are actually the standard natural amino acids. In fact, they're all modified in some way. Now, if you reduce that even further, you can see that on the right-hand side, you can reduce this to a single-letter peptide sequence representation. That is a sequence with which you can actually go now into UniProt, for example, in order to pull down content for cyclosporins. So the question is, what is right? What is a good representation for this molecule? Well, probably both things, but they are separated in terms of the knowledge base. And what I'm offering you today is tools with which you can actually bring these knowledge bases together, because we are kind of agnostic towards any of these representations. The nice picture you've seen on the bottom, this was kind of created by an application which is relatively new in our portfolio. We call it BioEddie. It's a JavaScript-based editor of molecules which are derived of biological sequences. And the first thing is it resembles somehow a chemical structure editor, so it is chemically aware. However, it's kind of creating a kind of reduced complexity view onto a molecule. A molecule is composed of individual monomers pulled from a library. The library contains the chemical descriptions of these monomers, and whenever you choose to kind of look at a specific part of your molecule structure, you can pull down the chemistry for this. Now, the 5 formats which are supported by this are mole format as a chemical structure format, Helm as a kind of industry initiative to create a standardized representation for biological molecules. This is a project driven by the Pistoia Alliance. We make sure that our applications actually natively support this standard representation. But you can also start from a sequence level and just pull in a sequence and then make utility of the fact that we're able to convert this back into a chemical structure, for example. Now, this is a very useful representation as long as you're dealing with relatively small biological molecules, say peptides, inhibitory RNAs, for example. We're able to kind of transform the view of that editor into things which are more convenient for, say, protein scientists. There's more interest in the actual sequence behind a given molecule. And then often on these sequences, you have a set of annotations which you might want to explore. The other thing is, which we've actually fairly recently only added, is as you leave the space of single proteins and you're looking at antibodies, for example, what you're often rather interested in is not necessarily a single monomer by monomer representation of your molecule, but you're looking at the architecture. You're just making one step backwards and say, okay, here I have a molecule which contains of my 2 light chains, 2 heavy chains. Within these chains, I have individual domains, the variable and constant regions of an antibody. Plus, if I chemically want to represent this with the bonds binding the domains together, then I can visualize these as well. They are part of my molecule representation, and hence I can use a fully expanded view of my antibody in order to push this through down to registration systems, for example. What you see now is a quick video, just seeing the tool a bit more live and in action. What I'm taking as an example here is a peptide hormone, oxytocin, and I'm starting off from the sequence level. I grab the sequence from somewhere, I can just copy and paste it there, trims all the white space, I hit load and I have my base sequence. Now, that's the base sequence of the peptide hormone, but it's not actually the ready, mature structure of oxytocin. What I'm showing here is that all of these views are somewhat also interactive, so you can start typing in the sequence and it's updating this on the canvas. Now, there is an amidation sitting on the C-terminal end of the peptide sequence, which I'm just pulling over from the library, and I draw a bond between these two monomers. And there's also a disulfide bridge, which kind of stabilizes structure a little more. There is some ambiguity in terms of selecting the proper attachment points. That's an information window which pops up. And now I have an accurate representation of mature oxytocin. I can choose to export this into, say, Helm format, and then I get that as a file. However, I can also start from the chemistry side of things. I'm using Marvin.js just to import oxytocin as a molecule. This is the structure of oxytocin. I export this from Marvin.js as a mol file. Actually, the step is just introduced at talking a bit about our naming technology. So then here, I'm selecting the mol format as an import format, and bam, there it is. It uses my library, which I have in order to interpret the chemical graph based on the content of my monomer library. And now, to make it clear, it is actually not relying on a fixed monomer library, but it's actually taking into account any custom monomer library you might have. This conversion is powered by a backend technology which we've developed, and it's yet another product is the Biomolecule Toolkit. So the conversion itself is not included in the thin client, but it's something which is provided by a web service API. The Biomolecule Toolkit as such, you can compare to JChem-Base. Essentially, it's a Java or REST web service API, which provides you with format representation IOs, the ability to standardize these molecule representations, have access to a data storage layer. We're now in the process of adding Oracle database support. We have so far MySQL support. There is built-in logic for registration of different entity types with different validation rules, all of this fully customizable. And then obviously, once you put this somewhere into a database, the value comes when you can ask very specific questions on what kind of molecular properties are present in a set of my molecules which I have stored in the database. This toolkit has now been brought as the backend for handling biological molecule information in a number of tools. Instant JChem is our own chemical data management tool. We have a plugin since August last year which provides you with the utility of using the Biomolecule Toolkit in combination with Instant JChem. Obviously, we want to move this in our Plexus Suite environment that's planned for the remainder of the year. But we also are seeing now, one step after the other, integration of our technology into other partner companies' product portfolios. Just to name Texelia Biosity, it's a small consulting company in Switzerland, and you'll hear more today from Jared about its integration into the eWorkbook suite from IDBS. Maybe some examples. So I've talked about this now as a single example in the beginning. What about if I took the whole content of Campbell? Now, Campbell was one of the pioneers actually adopting the Helm standard as an information tag for some of their molecules in order to serve a Helm representation with some of their molecules. The Campbell interface is what you see in the screenshot. And if you ask for what kind of queries can I ask that database, then mainly you can search this by a compound ID or by some attributes. A chemical structure search works in some ways, but it's fairly clunky. It's nice for chemical structure interrogation, but we might have other questions when we're looking at biomolecules, right? So what do? We grabbed the mol files for this kind of peptide content and started looking at what's the quality of their data and how can we benchmark our own tools. So we went and, well, looked at what are the contents of the mol files they are serving. There are a couple of standardization steps which we did. I named the tools which I used in order to go through that content on the bottom left. So I use KNIME as a workflow processor, and then a standardizer node in order to kind of create a representation which is readily digested then later on by our Biomolecule Toolkit. There was a couple of steps to be taken in order to bring this into a unique form. Then afterwards, I used the conversion utility from our BioToolkit in order to create a sequence-percepted molecular representation here in Helm format. And we've already identified at this stage that there are a couple of molecules which are kind of serving a wrong Helm representation because it's not considering the right connectivity between monomers. Here, for example, the usage of the regular L-glutamate versus the gamma-glutamate in a very short peptide. The second thing was then we can also canonicalize things, make sure that we perform a proper uniqueness check. Now again, this slide looks very complicated, but the essence is written on the left-hand side. So around 1,000 out of the 20,000 peptide notations, we had to change in order to make them truly unique. And following that, we also concluded that there are around 100 to 150 duplicates. It depends on whether you want to count the salt forms as individual substances or not. Last but not least, now we have that content essentially re-indexed. Now we can ask other types of questions which aren't supported by the chemical database by itself. So I can now ask for molecules containing oxytocin in the name fields. I can ask for, show me all molecules which have a similar sequence to oxytocin. How are you asking this in Campbell? It's not possible. I can also look deeper and say, okay, give me only those which contain non-natural amino acids, which contain a derivative of a very specific synthetic amino acid, for example. And I just give you here the number of hits, but in fact, you can get the full detail of these molecules back. A second use case, which we've actually rolled out to a customer, and Mike and Fred from the consultancy team took actually a lot of effort in order to make that happen. As you've heard in the morning session, we're using Instant JChem together with an add-on called MiniReg in order to offer especially small companies an environment where they can have chemical structure data management, a uniqueness check, the ability to register in batches or samples of these chemical structures, and then associate biological data to these structures. Now, why don't we do something very similar for biological substances? Because it's the same process, it's the same workflow. So we hooked in the Biomolecule Toolkit via a plugin into Instant JChem, make that integration work. And then we started rewiring the MiniReg application in order to meet the specifications of the customer. The customer wanted to register antibodies in pretty much the same way as you would expect to register small molecule structures. However, what's the specification of an antibody? That is, I only am defining an antibody by the variable part of the light chain, the variable part of the heavy chain, and essentially a text field which describes the scaffold onto which these domains are grafted. We have built into our toolkits the ability to essentially represent things where you have partial definition of a molecule. So this is one entity type you can define, and if that information is available, it's a valid molecule and you can register this and even do uniqueness checks. So what we delivered then was antibody definitions coming from an Excel file. Template could be registered using the information above. We did the duplication check and batch handling, assay data uploading in respect to the antibodies, and then the form views and reporting for exploring that data. What I want to convince you of with that is that we actually have the Biomolecule Toolkit as a backend technology together with BioEddie as essentially the sketching utility to be able to define such molecules as an end user in order to bring the capability of biomolecule management or molecule management into a variety of different applications. So we have that in Instant JChem. We're planning it for Plexus Connect. JChem for Office is on our radar. Other applications, you see a couple of examples now. So you've seen these screenshots. That's why I'm just skipping over them very briefly. That is about project management. This is a screenshot of the Texelia Biosity, essentially a data management and tracking system, including inventory for a small biochemistry lab. We're seeing here some screenshots. Jared will probably show a bit more up-to-date footage here, so I also just quickly skip over that. For those of you who are interested in where we want to take these products, we have these kind of sort of half-year plans. A lot of the things you see on the left-hand side, they're already semi-finished, so we're just in the process of closing a couple of these. We're still waiting for getting some refactors done in order to start pushing out workflow pipelining tools for KNIME and Pipeline Pilot. And then in the second half of the year, we're actually looking at, on one hand, providing a bit better antibody-drug conjugate support, which goes like beyond pure antibodies. There is a little complexity involved in how to handle the kind of graphical arrangement, molecule representation. And then on the toolkit side, we're kind of addressing facts which are very relevant for registration. The ability to kind of trace the genealogy of things which are connected to each other within a workflow. The ability to provide native support for batches of molecular entities I have stored in my database. And last but not least, a couple of things we have to do in our search capabilities. These are hot topics. There are a variety of different measures or metrics of similarity which we'd like to address based on sequence similarity, molecule similarity.
One question regarding the non-natural amino acids you presented. So how well is this library pre-populated in terms of non-natural amino acids? You took natural products and then chopped them apart and derived from there, or how did you get there, and how well it is populated?
We don't claim to provide a kind of well-curated and prepared monomer library. As a matter of fact, there's two things. There is the Pistoia Alliance Helm project. One of the major goals of that project is to come up with a publicly accepted monomer library which is as large as possible, so that everybody would not necessarily need to take care about migrating molecule descriptions in Helm between different monomer libraries. However, we are having tools which are essentially agnostic to the content of your monomer library. So a lot of organizations developing novel chemistry, they are very secretive for given reasons about some of their monomers, and they might not want to push these out to the public. So in these cases, we have to be prepared for this in any case. So at the moment, we would be able to pull in what is currently the kind of common accepted monomer set, which is part of the Pistoia Alliance effort. But we're also able to just pull in whatever you give us as monomer content.